Good morning, students. Today we are going to talk about antimicrobial resistance. So antimicrobial resistance is one of the most serious public health threats in the 21st century. Resistance to antimicrobial agents has become a major source of morbidity and mortality. So there are around 700,000 deaths worldwide. You can see that how big threat it is. Antimicrobial agents can be divided into groups based on the mechanism of antimicrobial activity. So first one is antibiotics, which inhibit the cell wall synthesis. The second one is depolarizing the cell membrane, inhibiting protein synthesis, inhibiting nuclei and synthesis. Inhibition of metabolic pathway in bacteria. Most pathogenic bacteria have the capability of developing resistance to at least some of the antimicrobial agents. So the factors which are contributing are environmental factors, drug-related, patient-related, and factors. The first one is environment-related factors: huge population and overcrowding, rapid spread, increased traveling, poor sanitation, increased community-acquired resistance, ineffective infection control program, increasing national and international travel, widespread use of antibiotics in animal husbandry and agriculture, and as medical cleansing products. Drug-related factors include fake drugs, quality of drugs, soaring use of antibiotics, overuse of antibiotics by purchasing them over the counter, irrational dose combination of antimicrobials, counterfeit and substandard drugs causing suboptimal blood clotting. Patient-related factors include poor adherence, poverty, lack of sanitation, lack of education, self-medication, and misconception. Physician-related are inappropriate use of available drugs, increased empiric polymicrobial use, overuse of microbials, inadequate dosing, lack of current knowledge and training. So doctors should have regular training. Clinicians should read their textbooks every day. So also just remember that it's natural that resistance of bacteria against antibiotics is okay, but it is not the fault of these four factors alone. So this gives you the idea about antibiotic resistance from the form of the table. So you can see that around 1 in 5 infections are caused by antimicrobial resistant organisms, and they are from the food and animals. So we'll talk what is bactericidal and what is bacteriostatic. Bacteriostatic antibiotics include chlorphenicol macrolides, clindamycin, sulfur, cotrimoxazole, that is cotrimoxazole, and tetracycline. Cidal drugs include aminoglycosides, acids, beta-lactams, vancomycins, quinolones, rifampicin, and antimicrobial resistance can be of two types: intrinsic and so intrinsic is always expressed in the species. It is the innate ability of the bacteria to resist a class of antibiotics. The most common bacterial mechanism involved in intrinsic resistance are reduced permeability to the outer membrane and natural activity of the efflux. Acquired resistance. We can say that this is a natural susceptibility to the microorganism which acquires by ways of not being affected by the drug. Bacteria acquires any genetic material by transformation, transposition, and conjugation, all terms horizontal gene transfer. Plus, the bacteria can experience mutation to its own chromosome. So, antibiotic resistance can be natural due to the lack of metabolic processes or target sites. Acquired can be either genetic or biochemical methods. According to genetic methods, it can be chromosomal by mutation, extrachromosomal methods by plasmids or between bacteria. Biochemical mechanisms may be by producing enzymes, preventing drug accumulation, modifying target sites, use alternate pathway, and quorum synthesis. Mechanism of antimicrobial resistance. Bacteria develop antimicrobial resistance by several mechanisms. The first one is by limiting the uptake of the drug, modification of drug target, inactivation of the drug, activation of efflux of a drug. Because of the difference in structure, there is variation in the types of mechanism used by gram-negative bacteria versus gram-positive bacteria. Gram-positive bacteria have very less commonly used uptake drug mechanisms and do not have the capacity for certain types of dry efflux mechanism, whereas gram-negative makes use of all four mechanisms respectively. So this picture shows the mechanisms of antimicrobial resistance. I request everyone to pause here and go through this slide. This is the most important slide of this chapter. Limiting uptake of drugs. There is a natural difference between the ability of the bacteria to limit the uptake of antimicrobial agents. The structure and function of the lipopolysaccharide layer achieved by gram-negative bacteria provides a barrier to certain bacteria modify the cell membrane porins, therefore preventing the antimicrobial from entering cells. There are two main ways by which porin changes can limit the uptake: a decrease in the number of porins present and the mutation that changes the selectivity of the porin. The strategy has been observed in many gram-negative bacteria such as Pseudomonas enterobacter and Klebsiella species against drugs like imipenem and aminoglycosides along with quinone. Gram-positive bacteria like Staphylococcus aureus recently developed resistance to vancomycin. One of the mechanisms has been used against vancomycin. Staph aureus produces thickening wall, which makes it difficult for the drug to enter into the cell and provides an intermediate resistance to vancomycin. These strains are designated as VASA, which is vancomycin intermediate Staphylococcus aureus. This is non-genetic modification of targets. There are multiple components in the bacterial cell wall that may be targeted by antimicrobial agents, and these are just as many targets as they can be modified by the bacteria to enable resistance to these. One mechanism of resistance of beta-lactam drugs are mostly by gram-positive bacteria through alteration of structure or number of penicillin-binding proteins. Penicillin-binding proteins are transcriptases involved in the construction of peptidoglycans in the cell. A change in the number of penicillin-binding proteins impacts the number of or the amount of drugs which can bind to the target. Okay, so the increase and decrease have influence on this factor. A change in structure of penicillin-binding protein, for example, penicillin-binding protein 2 in Staphylococcus aureus, which is acquisition by MEK gene, decreases the ability of the drug to bind or totally inhibit the drug binding. The glycoprotein, for example, vancomycin, work by inhibiting the cell wall synthesis, and lipoproteins such as daptomycin work by depolarizing the cell membrane. Resistance to vancomycin has become a major issue in the enterococci, that is VRE, vancomycin resistant enterococci, and Staphylococcus aureus, that is MRSA, methicillin resistant Staphylococcus aureus. Resistance is mediated through acquisition of VAN genes, which result in changing the structure of peptidoglycan precursors that cause decrease in the binding ability of the vancomycin. Daptomycin requires the presence of calcium binding. Mutation in gene, that is MPRF, changes the charges of the cell membrane surface to positive and inhibiting the binding nature of the calcium, therefore inhibiting or reducing the action of daptomycin. Resistance to drugs that target the ribosomal subunits may occur through ribosomal mutations. For example, in aminoglycosides, ribosomal subunits methylation, most commonly involving ER genes or ribosomal protection tetracyclines. These mechanisms interfere with the ability of the drug to bind to the ribosome. Selective toxicity of antibiotics that can affect only the bacterium, not human, due to the effect of specific genes that are present only in the bacterium. For drugs that target nucleic acid synthesis like fluoroquinolones, resistance through modification in DNA gyrase or topoisomerase can be seen. These mutations cause changes in the structure of gyrase and topoisomerase, which decrease or eliminate the ability of the drug to bind. Inactivation of the drug. There are two main ways which the bacteria inactivates the drug. First, by actual degradation of the drug, or second, by transforming or transfer of a chemical group to the drug. The first one, actual degradation of the drug, is done by beta-lactamases, which are a very large group of drug hydrolyzing enzymes. Another drug that can be inactivated by hydrolyzation is tetracycline by TET-X gene. Transfer of chemical groups to the drugs. Drug inactivation by transfer of chemical groups to the drugs mostly uses the transfer of acetyl group. There are a large number of transferases that have been identified. Acylation is the most diversely used mechanism and is known to be used against aminoglycosides. Active efflux pump. So bacteria possesses chromosomally encoded genes for efflux pumps. Some are expressed consecutively, and there are others which are induced by overexpression, which are acting under the certain environmental stimuli that when a suitable substrate is present. The drug pumps function primarily to rid the bacterial cell of the toxic substances and may have such effects, and many of these pumps will be transporting a large variety of compounds, for example bacterial resistant effluent. Most bacteria possess many different types of efflux pumps. There are 5 main families of efflux bacteria classified based on structure and energy sources. The first one is ATP binding cassette family, the multi-drug resistant and toxic compound extrusion family, the small multi-drug resistant family, the major facilitator subfamily, and resistance modulation cell division subfamily. All these details you will not get anywhere in the world, so you should read my lecture every day. Efflux found in gram-positive bacteria may confer intrinsic resistance because of being decoded or encoded on the chromosome. There are also gram-positive efflux forms known as carrier or plasmid. These pumps include members of MADE and MFS families. Efflux form found in gram-negative bacteria are widely distributed and may come from all 5 of the families, which mostly clinically significant belong to the R&D family. So the general structure of the efflux pumps is shown in this picture. So what is the difference between antibiotic and antimicrobial resistance? Antibiotic are medicines that prevent and treat the infections. Antibiotic resistance occurs when a bacteria change in response to the use of these medicines. Bacteria not human become antibiotic resistant. Okay, so bacteria become antibiotic resistance. There are bacteria which may then infect the humans and are harder to treat than the non-resistant bacteria. Antimicrobial resistance is a broader term encompassing resistance to drugs to treat infected people caused by other microbes as well as such parasites like malaria, viruses and fungi. So this is a gist. This is the second important slide which gives you a summary. I request every student of mine to pause the slide here, go through this, read it, understand it. If you have any queries, you can leave a post in the text box. And today I have got 1 million viewers. Thank you. Keep supporting.